so in the last few lectures, we talked about the reconstruction of metabolic networks and kind of gone through the 15-year history of development there. Turns out that the same thought process and similar workflows can be applied to reconstruct other cellular functions. And today we're going to talk about a few of these. So the outline of this lecture is as follows. We're going to talk a little bit about protein and protein properties. And some of these properties can be computed from the structure of proteins that are available, and these can now be formally put into reconstructions, a relatively new development, but opens up a lot of interesting possibilities. And we'll go through three examples of that today. Then we'll talk a little bit about the efforts that are undergoing to try to reconstruct the entire set of reactions that lead to protein synthesis. And this is often denoted by the E matrix or the expression matrix. So all the biochemical reactions that underlie the synthesis of a protein. And then we'll talk a little bit about how one couples together different networks in the cell, in particular metabolism and protein synthesis. And maybe we'll end by talking about some of the grander challenges associated with network reconstruction and integration to get an increasingly broad coverage of cellular functions in a target organism by getting in more and more of the functionally annotated genes into reconstruction. Okay, so let's first talk a little bit about protein. So as I think all of us know, protein catalyzes most reactions inside cells. And this icon indicates, in the top right there, indicates that reactions taking place in free solution proceed at a very different rate than on a protein surface. Basically, a protein surface has information about the right collision angle between the molecules to get the reaction to proceed more rapidly. And you know, somewhere in your past, you probably heard about the million-fold acceleration factors that enzymes possess in terms of catalyzing a reaction faster than it happens in solution. And some of these surface properties of protein are remarkably sophisticated. And I like this image. This is the RNA polymerase. That this may be one of the more complicated enzymes there is. So on the right image there, you see the helix, DNA helix in red, and it's being opened up and unwound. And you can see how the lagging strand, or the strand that's not being used as a template, is kind of wrapped around the enzyme and pulled along that groove. So the properties of the surface of protein are very intricate, and they kind of steer the chemistry that takes place inside cells. So based on the structures of protein, you can estimate or try to compute some of their properties. There are, of course, many properties of protein. Some are hard to compute, some are easier to compute. Probably none of them is easy to compute, but some of them are computable. So one of the properties that can be computed to a certain extent is the thermal stability of a protein. Other things relate to how a substrate fits into the active site of an enzyme, and you can maybe predict whether an enzyme can act on a substrate or not. And if it cannot, you might even be able to predict inhibitory actions of molecules that bind to the active pocket and block it. So there are many of these properties that can potentially be computed, and relatively few of these so far have been computed in the context of a network function of a protein. But we'll mention some of them in here. So there are three examples I'd like to go through that have been published that shows how mapping protein structure onto metabolic networks leads to productive results. The first one comes from the integration of the structural proteome of Thermatoca maritima that we talked about onto the metabolic reconstruction. So this is a relatively simple organism. We talked about it before, free-living thermophile. And there was a large-scale protein crystallization and structural determination effort undertaken to try to get the structures for a lot of the proteins that make up this organism. And it turns out that you can integrate these two very different worlds in biology, namely structural biology of protein and network biology. And you can actually analyze things. And that's what's indicated in this figure here. We have a genome. We can reconstruct the reaction network. We can clone the protein, crystallize them, and get their structures. Then we can actually associate the protein structures with the reactions in the network. Now, what sort of possibilities does this open up? So, when we're looking at an organism like Thermatoca, it's an evolved organism. It's developed pathways to do different things. And there are different models that have been proposed for pathway evolution. There's something called the patchwork model, shown on the left here, that assume there's a gene duplication, and an enzyme that acts on a metabolite learns how to act on its product, and then there's another duplication event, then slowly a pathway develops. The retrograde model that's shown on the left assumes that enzymes are promiscuous and kind of in the underground metabolism, as it's called now, in a given proteome, there are paths for a molecule to go through different reactions in this protein, but it's at a very low rate. But if a selection pressure is put on that property or that pathway, these enzymes may evolve to form an independent pathway. So by looking at the fold and the fold distribution of protein in Thermatoca pathways, there was clear evidence found in that analysis for the retrograde model. And I think we'll talk more about this later on, that there is a lot of chatter in metabolism. Enzymes are not as specific as we think they are, and they have the ability to convert molecules that are similar to their primary substrates, but at a very, very low rate. But that rate is malleable by evolution, and if there is selection pressure on that property, the protein can mutate and change and enhance the ability to carry out that reaction. So this is one of the things you can do when you can map protein structures onto a network, a reconstructed network. So let's then continue with this issue and ask the question, why are some enzymes highly specific and some are not? So if you read textbooks on biochemistry, like the one shown there, we are normally taught that enzymes are very powerful catalysts, highly specific, and highly efficient. Now, if you think about evolution and you go back in time, it's pretty clear that the ancestral enzymes were lazy and sloppy, so to speak. They were not very efficient, and they were not very specific. But with time, they evolved and got better at catalyzing certain reactions. But it turns out that many of modern enzymes are still non-specific and not that efficient. And that has raised a question that has been kind of latent in enzymology since a very famous paper in Science in the 70s. Why is this the case? Why is there only a subset of enzymes that really follows that first bullet point that is often stated in biochemistry textbooks? So it turns out that you can actually look at the GPRs in metabolic reconstructions and start to address this question. And this is a very detailed study here that appeared in Science a couple of years ago that we're not going to go through in detail. But the bottom line results of the paper are shown here. So we can now look at the GPRs and we can see how many reactions are actually associated with an enzyme. So you can classify the enzymes based on the GPRs into enzymes that are specific and are only known to catalyze one reaction, and more generalist enzymes that may be able to catalyze many reactions. And it turns out that if you study multiple different data sets, regulatory shifts, and a large number of disparate data types, you come to the conclusion that the enzymes that are highly specific and efficient are at certain pinch points in the metabolic network. And they tend to be under selection pressure. So to get the whole network to be very efficient to support growth or some other properties, those enzymes become very specific and very efficient. Every enzyme has a certain turnover rate, and a promiscuous enzyme will have to share that turnover rate between many reactions, whereas a specific enzyme uses the entire catalytic potential of an enzyme to catalyze a single reaction. That, if the flux through that reaction represents a selection pressure, you would expect that enzyme to become very specific and highly efficient. And that's what this study concluded. Now, the integration of a protein structure into a network can be very formal. So you can represent some mathematical properties of the enzyme as a part of the reconstruction. And one of the properties I mentioned earlier that we can compute is thermal stability. So you can incorporate as shown in the call-out there on the network. You can represent the binding pockets of an enzyme with respect to the reactants and the products of an enzyme. But what you can also do in here is you can associate the turnover rate of the enzyme to temperature. So we can calculate, predict, estimate the thermal sensitivity of the enzyme. So you can get basically Vmax as a function of temperature. So if you can do this on a genome scale, as was done in this study, you can actually specify the temperature in your simulation and then calculate all the relative Vmaxes, and then you can optimize the growth rate. Then you can dial in a different temperature and redo the calculation. And in this way, you can just increment, temperature, the value of temperature over a certain range, and then calculate the growth rate as a function of temperature. And remarkably, for E. coli, that works. And that is shown in the top right portion of this slide. So basically what's done here is that we can calculate the Vmax as a function of temperature, as I said. You can then vary the temperature and trace out this curve that shows growth rate as a function of temperature. And you see around the optimal growth rate of E. coli, which is body temperature, 37 degrees, the computed curve and the data matches very well. Now the computed curve will have a more restricted temperature range because you can't predict the thermal sensitivity over a wide range. So it's more restricted, as you can see, especially on the lower end of the range there. And then by looking at that curve, and these are methods we'll get introduced to later, you can estimate which pathways are limiting the growth at each temperature. And the pathway map shown below indicates in red the pathways that are fingered by such an analysis. And interestingly enough, many of them tend to be in the biosynthetic pathways of prosthetic groups, like vitamins. And it turns out that this observation has been made for other organisms, that one of the factors in, maybe a dominant factor or a prominent factor in the temperature sensitivity of organisms maps onto secondary metabolites like the prosthetic groups and their synthesis. So you see, if you can have protein structures, compute their properties, map them onto metabolic networks, you can do things that you never expected that you were able to do. And so the ability to have the protein, their properties, and their synthesis put into a network opens up a lot of different phenotypic properties that cannot be computed. So let me talk now then about the efforts that have been undertaken to formally reconstruct the process of protein synthesis, and then how it's integrated with a metabolic network. So I think, as was evident in the lecture about the E. coli reconstruction, it became clear shortly after 2000 that you could reconstruct biochemical reaction networks on a genome scale, and you could mathematically represent them, and you could get meaningful computation of phenotypic states. And Jenny Reed, who did the second version of the genome scale E. coli model, put together this review in 2003, shortly after this became clear. And that review lays out this vision shown in here, that metabolism isn't the only function in a cell that can be reconstructed in terms of its underlying reactions and stoichiometrically represented. The same thing should be true for transcription, translation, regulation, and any other process in a cell that you can parse out in terms of the underlying reactions. And what's also shown in this diagram to the left is the fact that if such a reconstruction could take place, it becomes a container or a context, or a container, let's use that word for now, into which you can map the omics data type simultaneously. And I think, as I mentioned earlier, the omics communities tend to be a little insular and are focused on their technologies and the data processing and so forth, but the need to integrate these data types is significant. And we'll talk more about that in the next lecture. Okay, now, so this vision was articulated in 2003, and it took almost 10 years to realize it. And here's an image from a paper that came out in 2009 that described the full reconstruction of translation and transcription in E. coli for every single open reading frame that you would include in the model. And in this diagram to the left, you see DNA there in pink. You see the RNA polymerase binding to it. You see then subsequently translation. You see the function of the chaperones that secure folding. You see at the bottom there in green and blue a protein complex being assembled, and then a prosthetic group being put into that complex. And that prosthetic group could be, vitamin 6, could be iron, or something like that. So this traces out from the gene and the sequence all the reactions to the formation of a fully functional complex. And that once that complex is fully functional, it gains the capability of carrying out the reaction. If it's incomplete in any way, it cannot carry out the reaction in a model. Now, with this are a lot of other processes that relate to functionalization of the molecules, and shown to the right of this diagram are the modifications of the rRNA and tRNA, and these reactions are pretty well known. A lot of chemical modification is needed of these molecules to get them to be fully functional. And of course, the same thing is true for the proteome itself. Protein get acylated, they get phosphorylated, and so forth. So there's a lot of detailing of the chemistry that these macromolecules carry out by these modifications. And that pentagon to the right illustrates what some of the data sources are that enabled this reconstruction. This took Ines Thiele, who was a student here at UCSD, over 2 years to do, to go through this process. And this was not a simple process. Now, once you have a reconstruction, there is always a number of things you can do with it. For this particular model, you can calculate modules, you can calculate the coherent function of components of the network and all of its states, so you can predict modularization, and it's largely consistent with some of the physical aggregates of protein that occur in this system. It was a clear container for omics data mapping, and it's hard for you to read the text on the top of this diagram, but the number of data types that you can map onto it are many more than anticipated in that 2003 paper, basically because those data types didn't exist. And then you can do some computation of physiological properties For instance, you can calculate the relative amount of protein to RNA as a function of growth rate. And it's well known that the RNA content in bacterial cells goes up relative to the protein content as the growth rate gets faster. And you can see in that bottom diagram, this reconstruction perfectly recapitulates that property. So that's an example of taking a completely new network and reconstructing it. Not easy. In this case, a lot of data available, but was achievable. So with a network like this being built, you can actually integrate it with metabolism. So the protein are needed to produce the metabolites are needed to build the protein, build the nucleic acids, and so forth. So this led to the construction of a new set of models that are called ME models for metabolism and expression. And the reconstructions and models that are based on them that we've been talking about are typically thought of as networks like shown on the left here that you may see in a textbook. But what we are going to be talking now about is a model that actually calculates the full proteome of a cell in addition to the metabolic network. And this starts to open up all sorts of possibilities like the three that I mentioned earlier. So let me just show you one thing that the ME matrix can do. So it can actually calculate the differential proteome under two different conditions. So you can calculate a graph like this. This looks like a differential expression plot. But this is a differential protein abundance plot calculated for two different growth states for thermotoca. So I think this is on two different sugars, I think arabinose and cellobiose. And you can see that most of the proteome is predicted to be used the same way. It's on that 45-degree line. But there are some protein that are clearly predicted to be differentially expressed. So these could be transporters and metabolic processes specific to that substrate and so forth. Now this, of course, can be directly tested experimentally, and you can just do the expression profile of a cell under these two conditions as a proxy for the proteome. Of course, if you had the full proteomic profile, you could use that too. And in this case, all the proteins that are predicted to be used differentially under these two growth conditions are actually seen to be differentially expressed in the transcriptome. So you actually, you predict the shifts in the proteome here. Now, since the transcriptome has to change, that means that there's some regulation that is acting there. And you can actually now look at the transcripts that go up and down. And because the genomic locations are in the reconstruction, like I've been mentioning before, you can now look at these promoters and look at what's similar between the promoters that are going up and down in this shift. And sure enough, a regulatory motif is found upstream from these genes that correspond to transcription factors binding there. And then what you can do with these data sets is you can detail them actually more. And what's shown in part 4 is this, in this diagram, is this discovery process that comes with this. Sometimes you see correction of the transcription unit architecture of the genome, and we'll talk much more about that in the next lecture. You can also get putative gene functions, like the transporter for one of the sugars was not known, and this gives you suggestions of which genes might encode that transcription factor. So this is becoming much more interesting, right? Here's a genome scale model that actually calculates the whole proteome of a cell and its function under two different conditions. Now, you can continue to reconstruct processes in a cell. And I put this one up because this reconstruction was done by Joanne Liu, who actually did this as a part of term project in this class. So she was interested in looking at the translocation processes in E. coli that move protein around. And basically, protein can be in the cytoplasm, they can be in the inner membrane, they can be in the periplasm, or they can be in the outer membrane. So they can be located in different places. And the middle of this slide shows the workflow that is followed and the different data types. So on the top left, you see the 5 predominant protein translocation processes in E. coli. In the middle of that panel, you see the assignment of these proteins into different compartments that's based on literature. And at the bottom, you see some kinetic constants that are indicate the activity of each one of these transporters. So to calculate the functional proteome, you have to know the activity level of a protein, because you have to balance the pathways, and the abundance of the protein is then computed based on its activity level. And virtually everything here that came out of the literature synchronized well. I think one of the turnover rates was found to have a unit error in it, a second rather than a minute. So when that was changed by a factor of 60, this whole system balanced pretty well. So as a result of that, there is now a me model that calculates, that has metabolism in it, as shown over here. It has the E matrix, the translation, shown to the bottom left. It has a protein translocation on the top right part. And what's indicated in this diagram here is that about 80% of the proteome is explicitly calculated with this model and this location. The little gray zone in there is the proteome that isn't accounted for. And now you can imagine that you can keep reconstructing these processes and putting it into the model. You know, multi-drug resistance things, the flagella, protein folding apparatus, and so forth. So you can systematically work your way to represent more and more of the proteome. So at the bottom of this slide, I think are some interesting explanations of what the difference between an M and a me model is. So I like the economic or the business interpretation of this. So an M model calculates for you the OpEx for running a particular phenotypic state. What are the operating costs associated with running that phenotypic state? The me model calculates the proteome as well, so it's also calculating the hardware. So it's also calculating the CapEx, you know, what you have to invest in before you can run that operating state. So a me model actually is an OpEx and a CapEx model for a cell. And as we'll see later in the class, this opens up all sorts of interesting possibilities in terms of computing limitations associated with the proteome, because protein aren't free at all. Okay, so that's kind of interesting. So this has gone reasonably well over the last decade or so. And you can imagine that based on this achievement and the comments that I made, that this process is going to go on until we cover more and more of cellular functions. So in this diagram, we see the metabolic network on the left, and to the bottom there we see, let's see where we see transcription and translation. I guess that's the top panel there in the middle. But we haven't reconstructed yet the transcriptional regulatory networks or the signaling networks. And those we'll talk more about in the next lecture, but the data types to do that are becoming available, although their mathematical representation, how you do the mathematical representation of them is not quite clear yet. I think the two-component systems in bacteria can be reconstructed now based on what we have, but the functions of the small RNA molecules we cannot, probably not reconstruct today, because we just don't know enough about their function. So here is the two-component system in E. coli. It's about 23 or 4 sensors in E. coli that have, in many cases, just a cylindrical, just a straight tie from sensing a signal and to the genome. But some of them are interacting as shown over there. And many of these reactions have already been tabulated and identified, and it is now possible to build this reconstruction and interface it with a me model. So finally, I just want to talk about signaling networks in mammalian cells, because there is so much interest in reconstructing them. And most of you have seen pathways like the one to the left of this diagram. There is some signal that's indicated by that lightning on top, and the initial signal may phosphorylate the protein A, and then that leads to phosphorylation of another protein B, and then another protein C, and that may lead to a translocation event, eventually leading to assembly of a transcriptional complex and a response in terms of gene expression. So pathways like this can be represented with a Boolean formalism, and there are a number of models of that type, as indicated there in the middle. They can also have a stoichiometric representation. If you know all the reactions, you can build a stoichiometric model for them. Now, it turns out that in spite of all the interest in reconstructing these networks, they're extremely hard to reconstruct. Basically because you cannot read out of the genome annotation the functionalities of these proteins and reconstruct a signaling network. Remember, we can do that for metabolism because we know those gene products so well. These networks are mostly reconstructed based on direct biochemical evidence. And it's very hard to do this on a genome scale because you don't know where your network starts and where it ends. And publications, even for well-known growth factors like EGF, if you look at them, the scope of these reconstructions is all different. So you can't compare the models, and it's not clear how they fit into the rest of the functions of a cell. So this represents a grand challenge, something that would be nice to be able to do, but the path here, the path forward here is not obvious. Okay, so let's make a few summary bullet points here at the end. So we're interested in expanding the reconstruction process beyond metabolism, and it turns out that properties of protein now can be computed from structure. We have a lot of structural information. Those properties can be mapped onto a network and included in computations, like the predicted Vmaxes or relative kcats as a function of temperature to predict the growth rate and how the growth rate changes. I skipped some of those, some of the figures in the text that show the workflows, but workflows are being developed to enable these reconstructions. They are somewhat similar to the workflows that have been developed for metabolism, but not identical, but they're similar. There is undoubtedly more work needed in developing and perfecting them. Probably more QC, QA steps needs to be included, but basic workflows are emerging for this process. So today, we can't do this for protein synthesis on a genome scale in bacteria. There is a reconstruction of the protein secretion pathway in yeast, all the way from the initial transcription event until the protein is secreted. There are efforts to do this also in human cells. Mammalian cells. Will we be able to expand this to calculate the full proteome in a mammalian cell? Who knows? Hopefully one day. We can now use protein localization pathways to localize protein in the different cellular compartments that we have, and I think that is important. I mentioned a few times in these lectures what the meaning of location in biology is, and one of them relates to the cellular compartment, so we can maybe compute that now. Signaling remains difficult. And if you choose to reconstruct these networks in terms of the detailed biochemistry, where you can get stoichiometric representation, you can integrate them in one step. And that's the power of the stoichiometric representation, is that you can integrate so easily multiple networks. So it's an extraordinarily scalable formalism. So that's a summary of the state of the art in reconstructing networks other than metabolism.